All right, so fatty acid degradation. This is going to occur in the mitochondria when the cell actually needs some energy. So this is typically degraded by a sequential removal of 2 carbon fragments at a time. If you remember, the length of the the common range for fatty acids were 12 carbons to 20 carbons. So they were even numbered carbons, and those are the most commonly found ones. And so if you break off 2 at a time, well, then you're not disrupting that, and you're going to end with a molecule that's probably 2 carbons long. This is known as beta oxidation because the beta carbon is oxidized. If you remember our display of our fatty acids, well, we saw the first carbon was an alpha carbon, the second carbon was a beta carbon. Now, acetyl-CoA is formed as the bond between the alpha and beta carbon atoms is actually broken. This process is common to saturated fatty acids of even numbers. Now, other mechanisms are employed to degrade unusual fatty acids, so odd chain fatty acids, branch chain fatty acids, or unsaturated fatty acids. Now, before beta oxidation can begin, each fatty acid is going to be activated, and that activation comes with, well, the production of that guy right there. So we have to have our fatty acid plus ATP, which we're going to hydrolyze, and coenzyme A. So because mitochondrial inner membrane is impermeable to most acyl-CoA, a carrier molecule called carnitine mediates the transport of acyl groups into the mitochondrial matrix. So if you ever drink any sort of energy drinks, one of the main ingredients, aside from taurine, one of the other main ingredients is carnitine. And so carnitine is what is going to help transport a fatty acid into the mitochondrial matrix. Now, sometimes beta oxidation of an acyl-CoA is going to follow this process. It's a 4-step cyclic process. And the first step is oxidation of activated fatty acid, hydration of a double bond, oxidation of a hydroxyl group, and thiolytic cleavage and release of acetyl-CoA. Each step is going to remove 2 carbons at a time from the carboxyl end of the fatty acid, and this repeats until all the carbons of the original saturated fatty acid are acetyl-CoA. This can be used in the citric acid cycle, as well as isoprenoid and ketone body synthesis. For the first step, we've got our activated fatty acid. We are going to add a double bond, and we're going to use FAD in order to do that, resulting in the production of FADH2. So one hydrogen atom from each of these carbons that are indicated here and here is going to be removed and transferred to our FAD. Here's going to be the result. Then we're going to have to hydrate that double bond. So this is going to be catalyzed by an enzyme known as hydrase, and water is going to be a cofactor. So what we're going to result in is we're going to be adding a hydroxide group and a hydrogen, where there previously were 2 hydrogens, then a double bond. Now we're going to replace that double bond with a OH and an H. So after that, we're going to oxidize that hydroxyl group. So the resulting hydroxyl group right there, this is going to be done by the utilization of a dehydrogenase enzyme, NAD+. We're going to end up with a double bonded oxygen and NADH. So now what we've done is we've made something that's almost symmetrical. If you look, we've got our double bonded oxygen there. And now what we can do is proceed from there. Okay, and this is where we're going to do our thiolytic cleavage. So acetyl-CoA molecule is going to be released as a product. Our enzyme is thiolase. The other product, acyl-CoA, now contains 2 fewer carbons. So what we've done is we've taken this molecule right here. We've got our kind of symmetrical almost C double bonded oxygen, and we've got an a coenzyme A that we're going to swoop in and basically put it right here and release this guy right here. So overall, the degradation of fatty acid requires 2 ATPs to activate. It's going to then generate 1 FADH2 and 1 NADH. So when you think about this in terms of you're removing 2 carbons at a time, okay, great. you have to invest 2 ATPs, but you're going to get 1 FADH2 and 1 NADH out of that. Now, for a saturated and an odd chain fatty acid, we're going to proceed as normal with even number fatty acids. So if you've got a chain that's 19 carbons long, you're going to go through breaking off 2 carbons at a time until you get to propionyl-CoA, which is a 3-carbon molecule. That propionyl-CoA is going to be converted to another molecule known as succinyl-CoA. This is going to require 1 ATP. An unsaturated fatty acid is going to be a little bit different. You're going to proceed as normal with your even number of fatty acids, or your even number of carbons. Your yield is going to be the exact same. This is going to require a couple of different enzymes, an isomerase and a reductase. An isomerase is going to rearrange a molecule to shift a double bond, and a reductase is going to reduce cis double bonds. You're going to require NADPH for that process. So when we think about the beta oxidation of a 18 carbon long fatty acid, well, you've got 18 carbons, you're going to invest 2 ATP, you're going to get 9 acetyl-CoA, 8 NADH, and 8 FADH2. Because if you remember, at each successive removal of 2 carbons, you're adding a new coenzyme A to make a new acetyl-CoA molecule. So 18 carbons breaks down into 9 acetyl-CoA molecules. 1 acetyl-CoA is going to yield, going through the citric acid cycle, 10 ATP, or ultimately, since you're doing 9 of them, you're going to yield 90 ATP from this. Each NADH is going to give you 2.5 ATP going through citric acid cycle, or going through the electron transport chain. So you're going to generate 20 ATP. 1 FADH2 at 1.5 ATP per molecule is going to give you 12 ATP. Minus the 2 ATP that you invested to get things kicked off, you're going to end up with 120 ATP. This circle is less dramatic when it's to the right of the answer. Instead, it's supposed to be over that. So you would have to oxidize more than 3 times the glucose molecules to get the same amount of energy. So in terms of what's a better, I guess, energy source, fats or sugars? Fats. You get a ton of energy. So of the acetyl-CoA that's produced during the fatty acid oxidation, most of it's going to go into citric acid cycle or isoprenoid synthesis. So it's ultimately going to go through the citric acid cycle to produce NADH, FADH2, so that you can extract more energy from that. Or it's going to be used for the synthesis of cholesterol or hormones or something like that. However, there always ends up some excess acetyl-CoA. That's going to be converted into ketone bodies in the mitochondria. Ketogenesis is a process in which acetyl-CoA is converted to acetoacetate, beta-hydroxybutyrate, and acetone. These are all examples of ketone bodies: acetoacetate, beta-hydroxybutyrate, and acetone. Acetone is the result of, or is a result of, ketosis. Now, skeletal and heart muscles can use ketone bodies for energy under normal conditions. Brain tissue cannot use those ketone bodies. However, under starved conditions, brain tissue can use ketone bodies for energy. Now, what this actually kind of looks like is, let's imagine that this is one of our acetyl-CoA molecules. That's going to be the enzyme acetoacetyl-CoA thiolase is going to produce acetoacetyl-CoA. HMG-CoA synthase is going to produce beta-hydroxymethylglutarol-CoA, or HMG-CoA. HMG-CoA lyase is going to produce acetoacetate. Acetoacetate is going to be broken down into acetone and beta-hydroxybutyrate, an example of a ketone body, or examples of ketone bodies here. So that's going to be kind of the fate of your acetyl-CoA that doesn't go through the citric acid cycle. But in an ideal situation, all of your acetyl-CoA is going to go either to isoprenoid biosynthesis or the citric acid cycle. All right, I'll see you in the next video.